Welcome back to the After School Project. This segment is about nucleic acids. There are two types of nucleic acids, namely the deoxyribose nucleic acid, also known as DNA, the ribonucleic acid, also known as RNA. Nucleic acids are made up of monomers or building blocks called nucleotides. Each nucleotide consists of a sugar portion, a phosphate portion, and a nitrogenous base. DNA molecules can be found in the nucleus, and the DNA found in the nucleus is called nuclear DNA. There's also mitochondrial DNA found in the mitochondria, and in plants, some DNA can be found in chloroplasts. In nature, the DNA molecule appears as a double helix, or a tightly wound ladder-like form. Each strand of the DNA molecule is made up of a sequence of DNA nucleotides. The following nitrogenous bases are found in a DNA molecule: adenine, thymine, cytosine, and guanine. Adenine always pairs with thymine, and cytosine always pairs with guanine. DNA functions in the makeup of genes which carry hereditary information. It also contains coded information for protein synthesis. In the interphase stage of cell division, the DNA molecule replicates. Replication is the duplication of the DNA molecule into two identical copies or replicas. In the process of DNA replication, the double helix structure unwinds, showing two strands of DNA. The weak hydrogen bonds between the nitrogenous bases break, and the two strands of DNA unzip. Each strand serves as a template on which a complement will be formed. Free-floating nucleotides in the cytoplasm build complementary DNA strands on each of the original strands, with cytosine always pairing with guanine and thymine always pairing with adenine. The enzyme DNA polymerase controls the joining of the nucleotides, and two identical copies are formed from the original DNA molecule. It is important for DNA to replicate before mitotic cellular division to ensure that the newly formed daughter cells have the same DNA molecule as the parent cell.